Part 1 of the Porphyrian notes for Honor Zoology. So first up, we're going to review some stuff from the last unit, reminding you that there are two types of animals, or two large groups. There's the vertebrates and the invertebrates. The vertebrates we'll get to later in the year, and those are animals with backbones, and invertebrates don't have a backbone, and we're going to spend the first 5 units of this course focusing, the next 4 units of this course focusing on invertebrates. About 97% of all animal species are actually invertebrates, which is why we're going to take so much time on them. All right, so for the phylum Porphyria is our first phylum we're going to look at. It is within the kingdom Animalia, just like all the phylums we're studying, or phyla we're studying. So the phylum Porphyria is the sponges. Their name, Porphyria, means pore bearers. They have a lot of pores or openings in them. As you can tell in this picture here of these sponges, there's obviously large holes in them. Right? There is about anywhere from 5,000 to 10,000 different species of sponges. They are the simplest multicellular animals that exist. Their cells are independent of each other, and they rely on water current for food, gas exchange, excretion, and reproduction. So a lot of those essential functions that we talked about in the last unit, this Porphyria relies on water current for those functions. Right? So most sponges are going to be marine, meaning they live in salt water. They're going to have tiny pores on the surface to allow water to enter and circulate through a series of canals where platonic organisms are filtered out and eaten. Now, platonic means plankton. So think of like the little plankton guy from SpongeBob. That SpongeBob would actually be eating plankton, just saying. So plankton or platonic describes small organisms that passively float or drift in bodies of water. These are 2 microscopic photos of some plankton. And then there's 2 types of plankton. There's zooplankton, which are technically animals, and then phytoplankton, which are technically a plant. We're not going to get too much into plankton. You just really need to know the definition of them, right? So back to sponges. Sponges are going to be sessile creatures, meaning they live attached to the bottom of the ocean or on a surface of something, and they're also filter feeders, so they're going to actively filter suspended food from the water. All right. For their anatomy, the outer surface is covered with flat cells and sometimes tube-like porous cells called ostia, which form canals to allow water to enter the sponge. And a lot of simple sponges will have one large atrium in the center of the sponge, which is called a spongicell. All right, and here's a picture of that spongicell, and it's normally has a big opening in the top of the sponge. So this is like we are looking like straight down in the sponge, and this is a rather large sponge. You can see there's a fish inside of him, but that is the spongicell, that large atrium in it. Right. So for the flow of water through it, it's going to have water pumped into the larger canal that's lined with what are called collar cells, and collar cells are also called choanocytes, and those have flagellum. Now, if you remember what flagellum is from your freshman biology class, they're an organelle that's like a little tail on the cell that like moves around to push the water through it. So that little, it's like a little tail whipping back and forth to push water through the organism or through the sponge's canals. So the collar cells, also called choanocytes, line the canals of the sponge, and they have flagellum on them to help push the water through. All right. So after the choanocytes, those are also going to help trap the foods in their collar, which is why they're called collar cells. So these little collars around the top are going to trap the food. And it's moved along towards the base of the collar, and then the food's then engulfed by that cell to form a food vacuole. So it basically like opens up and like the cell kind of surrounds it and encapsulates it, and then it's digested by enzymes, and there's some pH changes to break it down as well. And so then the digested food is passed on to amoeboid cells, and then it distributes to the other cells. So these collar cells trap the food. They take it in, they break it down, and then they kind of like one cell after the next kind of spread it around and share the nutrients from the food. All right. So you can kind of see here, this is a diagram of a simple sponge here. We have these like bluish green arrows are the flow of water through it. So they have these openings, and then you can see here these purple ones are the collar cells. They look like this close up. So you can see the flagellum that's going to push the water through. You have the little food particles are the dots, and they're going to get trapped in this collar. And then the vacuoles are these little things. So here's a vacuole getting started, is engulfing it, and then it's carrying it back, and then the amoeba site eventually gets it, and then it goes on to other cells past that. Okay. All right. For helping support the spongin, you're going to have a couple different things that are molecules of kind of like a molecule of sorts to help provide support. The first one is spongin. These are resistant fibers for support. And there's some microscopic pictures of them. And then some larger sponges may have what are called spicules, and they come in different shapes and sizes. And here's some microscopic photos of them. But they're like little sharp little spike things that help provide structure and support for the sponges. All right. Now let's talk about some different body forms of sponges. There's 3 main body forms we're going to see. The first one is the Ascon sponge. It's the simplest and least common body form of all sponges. These are considered simple sponges. They have porocytes that help regulate water circulation by contracting. And then they have ostia that are the outer openings of the porocytes and lead directly into a chamber called the spongicell, which is that atrium. They do have the choanocytes that use their flagellum to help move the water into the spongicell when then it exits out the osculum. So the water comes in from the side and then out through the top. All right, make sure for any test that we take, you do know not only the information here, but you can recognize that this picture is an Ascon sponge, or if I give you a picture like that you can relate the diagram to the name. All right. The second body type is the Sycon sponge. It's when a sponge walls appears folded. So basically we're taking that Ascon sponge and now folding in the sides. Water is still going to enter through the pores on the outside. Those pores are still going to contract, and then they lead into that spongicell. The choanocytes line the radial canal still, and the water will travel from the ostia through all the different canals into the spongicell. So it has a slightly longer path. As you can see here, it goes in through the pore, and then it has a little bit of more like length of the sponge to travel before it gets to that atrium. But overall, it's generally the same besides just that folded side. All right. All right, the last one is the Leucon sponge. They have an extensive branch canal system, and they're considered complex sponges, even though they're really not that complex because, let's face it, it's a sponge, right? Water will enter the sponge through the ostia and move through branched incurrent canals, which lead to choanocytes, or choanocyte-lined chambers, rather. The canals lead away from the chambers, and they're called excurrent canals. They do not have a spongicell, so there's no atrium in these sponges. They do have multiple exit points called oscula for water leaving the sponge, and they have an increased surface area, and they can filter very large volumes of water. All right, and this is a picture of that. So you can see there's no main atrium like the other 2 types. And water, there's a lot of different pores that water can come in through, but then some pores that are generally kind of larger are going to be where the water exits. All right, so the water always goes through the same pores and exits out the same pores. All right, and that is it for part 1 of the sponge notes. Make sure that you watch part 2 as well.